So we were very glad to present our genomic cohort from the Dana-Farber Cancer Institute during the HER2-low session of San Antonio. And what we did was looking at an in-house NGS panel called OncoPanel that we do in most of the patients with metastatic breast cancer at Dana-Farber in 1,039 patients with HER2 non-amplified breast cancer, dividing them HER2-low compared to HER2-0, and also depending on hormone receptor status. And what we found was that there were some mutations that seemed to be enriched in HER2-0, like TP53 and NF1, some other enriched in instead HER2-low, like MTOR. But in the end, when you correct for multiple, for false discovery rate, there was really nothing that was significantly differently distributed in HER2-low versus HER2-0 when you correct for hormone receptor status. That is always very important. And I think this tells us something that we're seeing over and over, that HER2-low and HER2-0 are not really different molecular entities. There are slight phenotypic differences that we're studying and trying to understand, but in general there is not a strong distinction, and probably immunohistochemistry is not really the best way to dissect HER2-0 from HER2-low. And so basically I would say that our study shows that genomically HER2-low is not very different from HER2-0, and I was glad to see that also other studies supported this and were presented also at San Antonio. And overall I think we're going in this direction, thinking that HER2-low is not a distinct molecular entity, despite it can be targeted with antibody-drug conjugates.